So determination of the nucleic acid type. On the basis of the nucleic acid, of course, we know now that we have different kinds of the nucleic acid. There are RNAs, ribonucleic acid, or there is the deoxyribonucleic acid. There may be the single stranded or there may be the double stranded, both of them. So we can just determine the nucleic acid type on the basis of the nucleic acid inhibitors. What are the nucleic acid types? We already know that we have the deoxyribonucleic acid, we have the ribonucleic acid, and both of them may be the single stranded or the double stranded of them. Double stranded DNAs, double stranded deoxyribonucleic acid, or the positive sense single stranded RNA. We have the different viruses which are positive sense single stranded RNA. We have already seen the classification of the viruses. Negative sense single stranded RNAs is there, and then the double stranded RNA is already present in that nucleic acids. So on the basis of the nucleic acid, we use the nucleic acid inhibitors, for which on the basis of which we can see the different determination of the nucleic acid inside the body or where inside the laboratory. Use of the DNA inhibitors determine the type of the nucleic acid a virus possesses. On the basis of the nucleic acid, we can just determine, because we have already seen different kind of the viruses. Some of them are the DNA viruses, some of them the RNA viruses. Therefore, on the basis of the DNA or on the basis of the nucleic acid residing inside the virus, we can just isolate the different, we can differentiate the different kind of the virus, and we can diagnose a lot of diseases on the basis of the nucleic acids. So determination of the nucleic acid. The most important thing for the determination of the nucleic acid are the DNA inhibitors. These DNA inhibitors are already present, and then we can have different kind of the DNA inhibitors. For example, 5-fluoro-2-deoxyuridinase is the DNA inhibitor. It is also called FUDR. And then 5-bromo-2-deoxyuridine, it is called BUDR. And then 5-iodo-2-deoxyuridine is present, which is also called an IUDR. So these are different kind of the DNA inhibitors, on the basis of which we will put these DNA inhibitors in the reaction, and on the basis of the reaction we can isolate that what kind of the virus is this. Action of the DNA inhibitors. Specific inhibition of the viral DNA when added to the cell culture. Of course, when we add the cell culture, then the specific reaction will act upon, and on the basis of which we need to determine that which kind of the nucleic acid is living inside the virus. Just like interpretation, if the growth of course we have the DNA inhibitors, so if the growth is inhibited, then it is understood that the growth of inhibitor, and we are going to add the inhibitors. So it is presumed that the virus will contain DNA because it has inhibited the DNA. We have already put the DNA inhibitor inside it. And if the growth is not inhibited, we are using the inhibitors, so it is clear that it will be the RNA virus.